targeting aptamer work. Before I start to talk about those aptamer, I would like to, I guess the audience already know about G quadruplex, but I just give a very brief introduction so that I can focus more on the data part. So G-rich sequence in DNA and RNA can fold into G quartet, like as shown in here. And a few of them stacked on top of each other, stabilized by the potassium ion to form the G quadruplex. And many recent transcriptome-wise study, including some from our lab, have showed that besides this canonical sequence, canonical G quadruplex, there are many other types of G quadruplex that can be formed using different types of sequence. I like to call them like structural subtypes of G quadruplex. And there are many studies that shows individual cases of G quadruplex functional, and they're responsible for different types of biological process. Here, I'm just showing you a few of them here: replication, transcription, translation, but they do much more than that. And this is an aptamer talk, so I'm not going to go deep into the biology and mechanism. But we have found many functional and important G quadruplex over the years, and other groups have also demonstrated that. And you can find some of them from the review and some publication. So I want to get more into why what inspired us or what motivates us to do this project or start this project. So we want to have some tools that can target G quadruplexes. Some of you here are expert in aptamer and G quadruplex as well. So you probably have known there are many small molecules that can bind to G quadruplex, such as some shown in here, PDS, developed by Shankar Balasubramanian's lab, which I did my postdoc with, BRCA19, FANDC3. There's also antibody, nanobody that have developed peptides, oligonucleotide as well. So these are good, useful tools, but they also have some limitation in terms of selectivity. They do bind quite well, like nanomolar or some even picomolar level, but they do have some limitation in selectivity. So at that time, when I started my group in 2016, so I asked the question, so can we develop L-aptamers to do this? So some important notes, and we'll come back to these points later on, is so the naturally occurring RNA is in the D form, so it's a chiral molecule, biomolecule. The unnatural form is L form, shown in here. So it's just the mirror image of each other. So because of that, they have same physical properties. As I said, it's unnatural, so they will be resistant to nucleus digestion. So this is one good properties to have as an aptamer when we build aptamer later on. Because it's unnatural and people have already studied some lRNA in the older days, they show that they actually cannot form contiguous Watson-Crick base pairing with the natural dRNA. So if we can build aptamer based on this, so the recognition mode or recognition element will most likely be due to the non-Watson-Crick base pairing or even tertiary interaction. So this might give us more specific recognition, and that's part of the reason why we want to try to see if we can develop L-aptamer as additional tool sets for G quadruplex targeting. So we just ask some simple question to start with to test the feasibility. So can it be used to target non-canonical structures such as G4? So I'm trying to phrase it non-canonical here because often-wise, when we talk about DNA, RNA structure, it's either the double helical structure, stem loops, or hairpins, something like that. G quadruplex is sort of the outsider or the oddball. So we want to see if L-aptamer can be used to target non-canonical structures such as G4. If so, can we use it to distinguish between non-G4s versus G4? So for example, hairpin versus G4. And I will show you some data later on where we can push slightly more to distinguish maybe into potentially individual RG4 specific one, because I've shown you there are many structural subtypes, right? And if so, can it be used to interfere with G4 associated interaction and control gene function as well? Okay, so this project has started for a few years. I'm not going to go through all of them in this 15 minutes. So just to a quick sum up of some of the early work that we did. So we have some RA and PhD who have started off the work and developed the platform we call RG4 It's published for the early work and the protocol is in Nature protocol. So you can talk to me or you can look at the paper if you are interested in this platform after the talk. So today I'm going to focus on one particular L-aptamer and the target that we use as is an important G4 that we found in the APP, so amino precursor protein. So APP is important because that the proteolysis of the APP will cause the A beta to form and the A beta will aggregate and they have been found in the brains of Alzheimer's disease patients. So if we can have ways to regulate or manipulate the A beta production or APP production, then we may have ways to prevent or slow down or potentially cure Alzheimer's disease. So that's the intention. This part of the work is done when I was a PhD. So we found actually there's a 3' UTR-RG4 in there, relatively conserved, 4 runs of Gs and interspace with the loop sequence. Just some standard G-quadruplex assay. We did CD to show that it's dependent on potassium. So this gives some indication that this forms RG4. So that's our target, okay? So another PhD in my lab, Kai-Xin, have done some assays to verify and to provide more structural information of the target. So then when we design the L-aptamer or to do the L-aptamer, we will have more information to start with. I'm not going to go through the data in this slide, just to show you if you have some RNA that potentially contains G-quadruplex, you can use this assay and they're all published. I'll skip this slide. This slide is just to show you that the RG4 is important for the function. It basically will affect the translation. The presence of RG4 will affect the translation. We use a reporter gene system to demonstrate that, and we found that by manipulating the thermal stability of G4, we can play around with the translation. Okay, so these slides are just to show you that these RG4 targets presence and what kind of biological role that they do, and it's paved the way of what we are trying to develop now. So Haizhou and other PhD in my lab used the RG4 Silex platform. We managed to develop aptamer structure as shown here. We call it aptamer 8. It's just number 8 from the NGS sequence readout that we get. No, the Sanger sequencing readout. Danyang will talk about the NGS in the next talk. So we have this aptamer. So as some of the speakers already mentioned, so the structure is important, but of course we also want to test different things. So we sort of permutate and mutate and substitute. We are just showing 2 examples here, 8E, 8F. Obviously we do 8A to 8D and etc. So when we have 8E shown in here, so this doesn't bind. 8F, it binds. So this basically have shortened the aptamer 8 from, I don't know, 30 something, 39 nucleotide down to 33 nucleotide in here. KD is shown in here, 49 nanomolar, which is acceptable, not very great, but I think it's reasonable. And I didn't have time to cover that, but based on what we have done in the past from the first 2 L-aptamer work, we found that actually many of the L-aptamer that we developed later on actually contains the G-quadruplex as well. So it's our target is G-quadruplex. Our L-aptamer also contains a G-quadruplex. We don't have the 3D structure to show you today, but it's most likely there's some form of stacking plus some form of interaction that provides this affinity and specificity. So because of that, so we also did some of the standard G4 characterization assay to showcase that the aptamer 8F contains a G-quadruplex. So this is CD assay, again, potassium dependent, UV melting at 295 nanometer, have a hypochromic shift. So that's an indication of G4 formation. Some NMM, THT, these are G4 ligand that binds to G4, and when you bind, it will enhance the fluorescence. So these 4 assays both confirm that the 8F actually contains the G quadruplex. And the reason why you don't see sort of that structure in here is this is done by M4. So M4 doesn't allow that functionality. They do not predict G4 in there. So you have to be careful if whenever you work with aptamer, if potentially that have G-rich sequence, and you might need to do some of these assays in here. We do some mutations. I think, again, some speaker earlier today also mentioned this is important to do some form of mutation, covariation, just to confirm whether it's the structure or the sequence that's important for the binding. So long story short, just looking at the MSA gel in here. So some of them which are in red are some important nucleotide that are responsible for the binding. M3, M11, M8 in here. Some are less affected by the mutation. So some of these in greens. We also do some double mutation here to make some of them more important. Like M4 is a double mutation for M12, M13, something like that in here. So we do find some nucleotides that are important. And again, I don't have the 3D structure. I do want to have it to tell you more, hopefully next year. So based on this, we found the nucleotides that are important for the binding. And then the ones that I'm showing you here, we use the D aptamer and L target to do it because it's cheaper to do it this way. And because, as I said to you, this is mirror image. So the things that happens for D aptamer binding to L target should reflect equally well with the L aptamer versus the D target. So this is why we do many of the characterization using the D aptamer form. So then the remaining style are all on L aptamer. So we purchase chemically synthesized L aptamer and then bind to the D target, the DAPP RG4 target. And we found the KD to be quite similar to the L aptamer, D aptamer L target one. So this is 24 nanomolar. And again, I think some of the speaker already mentioned magnesium sometimes is quite important. So we do test that, especially for this case, right? It's not contiguous Watson-Crick base strength. It's probably due to non-Watson-Crick or tertiary interaction. And many of the times magnesium is very important for this type of interaction. So we test that and we found it's magnesium ion dependent. We also tested under lithium condition and we found the binding is weak. So, but for this case, it's not definitive because both our target and the aptamer both contains G quadruplex. So we don't know whether it's probably affecting both of the structure fold in there. One of the important things that we start with, right? So we want to see the selectivity. So we have this DNA, RNA hairpins in the lab. So we test it. So there's no gel shift, suggesting that these aptamer do not bind to these motifs. We have some poly A, poly C, poly U, they don't bind. We have many of the DNA G4 and some of these RNA G4 here also doesn't bind. Obviously, you do see some binding on some other RG4 in here. Mutated APP, we don't have binding. So this APP RG4 is our intended target. So the binding is also shown. So you can see there's some promising potential in terms of achieving some selectivity, but we are not quite there yet to get to the individual RG4 specificity that I was trying to get. But I think it's a good start because many of the small molecule or antibody, they do bind to all the RG4 and all the DG4. They only show specificity versus the hairpin or the single strand or random coil RNA. So I think this is not bad. At last, we try to demonstrate this aptamer. When we add to the cell, it can showcase some regulation in gene expression. So we start off with some reporter genes, so where we have the RG4 and mutated RG4 target, the APP ones. And then we titrate in either the L aptamer or we have the D aptamer as the control. And what we found that is in the wild type construct, we do see that the L-aptamer addition will suppress the translation, meaning that probably the L-aptamer bind to it and stabilize or either block the translation process. And we don't see that for the D-aptamer control. We also do it for the mutant version, and we don't see any significant differences in it. To go one step further to look into the APP protein, we actually transfect APP plasmid, full ORF and full 3' UTR APP together also with a MIG-TAC just for our detection purpose, and do the Western blot after titrating with the L-aptamer. And we found that the APP protein reduced when we add in more of the L-aptamer, same with the MIG-TAC as well, just to further verifying that this is from the plasmid itself. Okay, so this is where we are right now. And obviously we're developing even more other types of aptamer. So I will wait for the Q&A section maybe. So I hope I've shown you today that L-aptamer can be used to target non-canonical G-quadruplex, non-canonical structures such as G4. It can distinguish between RG4 and happy in random non-G4 stuff. Also okay for some DG4 and even some RG4. We're not quite there yet for individual specific RG4, but I think building on this framework, we can further modify and if we can get the 3D structure, it would be much easier to know where to mutate further. I've shown you, not in this talk, we didn't show you the G4 protein interaction that the L-aptamer can interfere, but I've shown you it can suppress the translation by targeting the APP 3' UTR. So overall, we have developed a platform. So again, feel free to use it. The protocol is published. And we have shown you the L-aptamer can bind to biological relevant RG4 with reasonable affinity and specificity. And this aptamer can be used for many different applications, and we are currently exploring that in different targets and different applications. And I think Danyang will also tell a little bit more on her target later on. Okay, so with that, I would like to thank first the organizer for the talk, and also my lab before pandemic and after pandemic sort of, and my collaborator, funding agency. And if you're interested in the work that we are doing, we are always recruiting, so feel free to email me or talk to me after the talk. So thank you.